Hello, everyone. Thank you so much for being flexible and letting me do the second part of our kind of growth and metabolism lecture online. I had a appointment that I just could not reschedule, unfortunately. So I really, really do appreciate your flexibility. This will be a one-time thing, but I'm very appreciative to all of you for your willingness to help me out here. So we're going to go through metabolism here in chapter 8. I really hope that a lot of this will be review for you, but I struggled to find the point where I thought, old and new knowledge kind of would take over. So if you're finding, hey, I think I know like 50% or more of this, that's fantastic. Really, from the intro bio threshold, even you should know over half of this. But sometimes I understand too that that might have been a little while ago for you, or it might just have been something that you didn't fully understand the first time. So I think it's okay here to have a little bit more of that refresher. But just so you know that that's kind of where I would think you would be at, would be that you would know the majority of the first part of this section. So when we talk about metabolism, we could talk about metabolism in any cell, and we would say that it's all of the physical and chemical workings of that cell. It's all of the combined reactions and processes coming together. And we would define that metabolism as two major types of chemical reactions. We would say they are the catabolic reactions, those that are degrading, breaking materials down, taking a large molecule, and from that large molecule, breaking it down to those smaller molecules, with the goal here being to release energy. And now we do this all the time. We take in large, complex molecules, we take in sugars and fats and proteins, and we break them down in a catabolic process to release energy, to get something usable from it. In anabolism, we're looking at a biosynthesis process. So how we can build up macromolecules from smaller molecules. So how do I build a polypeptide in a more complex protein? How do I string lipids together? How do I string nucleic acids together? How do I have photosynthesis in organisms like our cyanobacteria and our purple and green sulfur bacteria? How do these processes occur? And they occur in anabolic nature to build things up. And that building requires energy be added to those procedures, to those steps. Those building steps are facilitated by enzymes. And you know by now that enzymes are chemical catalysts, biological catalysts in your cells and the cells of microbes, that reactions do not proceed without enzymes. Enzymes speed up reactions in a number of different mechanisms, but often by promoting better positioning of the different reactants that are entering into that reaction, or reactant that's going into that reaction, to position things in such a way that combinations or breaking of bonds is more feasible. So how can it apply strain if a bond is to be broken? How can it better position if two things are about to be united together? Enzymes, we remember that they are reusable. They usually are reused many times before the cell has to recycle them into their working parts. And they are, again, just incredibly important. Without enzymes, life as we know it would not proceed. It would not exist. And this is true of microbial life as well. Now, when you learned about enzymes, you probably first started by learning about a simple enzyme, one that has a protein, and that protein lowered the activation energy and a reaction magically proceeded faster. And as you got further in biology, you added in other components. So you added in things like coenzymes. And hopefully you learned that there's a difference between things that are called coenzymes and things that are called cofactors. So coenzymes are these organic factors that are not protein, but they are organic in nature. And they may serve as these temporary carriers for molecules and substances. Vitamins commonly serve this role as coenzymes. And when you have this coenzyme portion, you are bringing together this protein and this non-protein molecule to have this completed whole, which we call the conjugated enzyme. So the other part here then is the apoenzyme. So the apoenzyme is just the protein part by itself. So if we look at the picture here we have what I think of as a simple enzyme, one enzyme by itself, no additional factors are needed. Here I have an apoenzyme where I've added a coenzyme, and now together they have formed this conjugated enzyme structure. And then here I'm going to add in another component. This one says I'm adding in a metallic factor. So usually when we have something like a metallic factor, we're referring to a cofactor. So metallic cofactors are going to help to activate an enzyme. They may bring the active site and the substrate together or may help with that process. And they typically participate in that chemical reaction directly by facilitating that enzyme substrate binding. So by bringing those two components together. Now, there are a number of different enzymes that we see in microbes. There's a short list of them here. But notice here we have a wealth of different metallic cofactors that all can play a role and a number of different processes here that are all critical to life. So how do enzymes help us synthesize DNA? How do they help us break down harmful materials like hydrogen peroxide? How do we add phosphate to different molecules? All these things that might be part of the Krebs cycle or synthesis of, any complex organic material is going to involve an enzyme. It's difficult to list things because it's everything. So in an enzyme, we remember that an enzyme interaction, we know we have an enzyme substrate interaction that requires the use of the active site. So when we have what's called an induced fit, the substrate has this this perfect tight bond with its active site. And we know that active sites are typically specific to one individual substrate that can occupy that spot. After interaction to form what would be the enzyme substrate complex, products are then released after the reaction here is complete. So we have examples here then where we have substrates going in that have perfect fit, other substrates or other chemicals, other molecules in the cell cannot fit. They do not. Again, products are produced. And here we've got just another similar example showing us then how that process of fit is working. Enzymes have a number of different actions that they can use. So in microbes, we think about exoenzymes and we think about endoenzymes. So exoenzymes are enzymes that are secreted or transported outside of the cell, and outside of the cell, they're used to break down things that are going to be beneficial or things that are going to, or maybe are harmful. So they may break down something like a large food molecule to help then bring that inside. We see that a lot in microbiology. So break down food outside of the cell and then let it then transport it in. Or it might be used for to break down a harmful chemical. So to detoxify something, make it safe. So cellulase and amylase might be used to break down the cellulose and starch of a food source, and penicillinase could be used to break down penicillin, which could potentially harm that bacteria and make it, that example, of course, we'd have resistance then to penicillin. An endoenzyme then is most common, so it's what most enzymes are. They're enzymes that actually act within the cell. So they are retained intracellularly, they function inside of the cell, and every process that a cell has to do, whether that's to break down food or to build up a nucleic acid, to do the Krebs cycle, to do like glycolysis or whatever it is, any process you can think of that's inside of a cell is going to use an endoenzyme. Built off of those two, we also have enzymes that are constitutive enzymes and those that are regulated enzymes. So constitutive enzymes are always produced. They are constantly present. So what types of materials do we always have to be building and synthesizing in cells? What processes are constantly occurring? A lot of times here we think about metabolism, we think about glycolysis, we think about things that are just constantly in need, so they must always be produced. Now, a constitutive enzyme is advantageous because it is always going to be available. If you need it, it is there. Now, what that also means, though, is let's say that the enzyme is not particularly needed at that moment. Well, then maybe you're producing something you don't need. But you have to assume that it's constitutively expressed, it's produced all the time because that's what the cell needs. It's not doing it in a wasteful way. Now, a regulated enzyme is induced or repressed because the cell needs it and doesn't need it at different times. So it's important here that we have the ability to turn it on. We can induce it, we can turn it off, we can repress it as we have changes in our environment and as we have changes to things like different substrates that are available. So we also see a lot of this in metabolism within bacteria. So let's say that we are looking at something like the presence of the sugar lactose. And we want to break down lactose, but only when it's available and only when we need to use it, because better sugars, those that are easier to break down, are not there. So we have this system that can be induced, or we have a system that can be repressed. We also have a number of other things like the tryptophan operon and other groups of genes that are regulated in this way so we can maximize our resources. They're advantageous because you don't waste, but they can be disadvantageous because they tend to be slower. It takes a while to accumulate that level, so it's not ready at a moment's notice. There has to be that little lag in between. Cells are really fast though. Sorry for not clicking this up faster. Cells are really fast though, so these probably isn't that much of a huge issue. So here again is a constitutive enzyme. Notice here it doesn't matter how much substrate is present for the reaction because it's always going to have enzyme. And notice here that we've got an example where a substrate becomes more plentiful, so the enzyme becomes more plentiful, but this is induced. And here we have an example where a substrate is lost or it's no longer present, and so we no longer have a need to make a bunch of that enzyme because it would be wasteful. In these different processes that we talk about, and we talk about enzymes helping to facilitate building up and breaking down molecules during these catabolic and anabolic processes, we're usually referring back to this really core group of reactions, which are condensation and dehydration reactions. Now, if you have condensation on your front lawn, you have dew on your front lawn or condensation on the side of a glass, you know that's because water is kind of precipitating out, right? It's forming large droplets. Okay, same thing happens in a condensation reaction. So here what we do is we form bonds, and doing so by forming the bond here between this carbon and this carbon here off of this maltose, we link them here by this oxygen, we pull out a water molecule. So pull out a water molecule, water condenses, and there's a bond here that's formed. Now a dehydration reaction then, so if you become dehydrated, it's because the water has been taken out. So here water is being taken out of where it is and is brought into this brought into this reaction. So the enzyme here is using the OH and the H groups, and it's using those to link then these together. Link these to these AA1 and AA2, allowing them to separate. My brain thought that this was connected, but I looked at it for half a second, then I went, wait, no, that's not right, pause. So by adding these groups, adding these new kind of cap, almost think of it like a cap here that's being added, they're allowed to separate. So again, condensation, water is being removed from these and they're being linked together, and dehydration, water is being added in and they're being split apart. We probably have also heard this as a hydrolysis reaction. So adding a water molecule to break. Which of the following statements about enzymes is correct? They show no substrate specificity. They are consumed by the reaction. They may be active extracellularly. They are composed only of protein. So pause if you need a second to think. And I'll pause as well. The correct statement here is they may be active extracellularly. So we know that they are highly specific. They are not consumed by reactions. They are reusable, and they are built from protein often, but there are other components, including those cofactors and coenzymes. So coenzymes there being those other organic materials, cofactors being those often metallic materials that improved the enzyme or better facilitated the reaction. Okay, next is sensitivity of enzymes to their environment. So enzymes are incredibly sensitive to their environment. They require a set group of conditions in order to function optimally. So they have a set pH, temperature, osmotic pressure, and when conditions shift away from those, enzymes may become very, very slow, or they may break down completely. So a great example is temperature. So at lower temperatures, an enzyme may become kind of sluggish, and at high temperatures, because it's a protein, the enzyme may break down, or it's predominantly a protein. The enzyme may break down. So we get also with that then those enzymes that may become unstable. We call that a labile enzyme. Or again, as I mentioned, they may denature, they may break down because they are weakened. And again, at temperature is a great example of that. Using a microbial example, explain how free-living microbial cells may struggle to maintain these conditions compared to human cells. So a lot of people in the class are geared towards that idea of humans. So let's make a comparison here. Human cells are constantly under regulation. We are constantly under regulation to maintain homeostasis. How can we maintain set temperatures, pHs, and pressures? Although they may differ over our body, but those areas, those regions stay the same. My stomach pH should always be within a tight range. My intestinal pH should always be in a tight range. My blood pH and temperature and all those factors should be within a tight range, which means the cells and the enzymes of those cells are going to be kept in those conditions. Now, in contrast, think about an enzyme that's within a microbe. What if you're an aquatic microbe and you're swimming through the oceans, using maybe you got little cilia and you're moving through the oceans or flagella, and it's cold and then it's warmer, it's deeper in some places, there's more and less pressure. Maybe the water channel has different pHs in different regions because there's differences in oxygen concentration, other things. You have to deal with all those factors. And so we may see that those reactions then have a little bit more fluctuation that they're going to undergo, a little bit more, again, difficulty because of their environments. Now, some microbes probably will maintain pretty solid conditions, but microbes, again, as simple cells, often don't have, I shouldn't say all microbes, many microbes, bacteria are key, often are going to find themselves either in harsh conditions or in variable conditions. And so we have to have, kind of that little bit of forethought then as well. Just something to kind of think about for those of you who are thinking about human related ideas, how it might compare. So another area that we can think about with enzymes is the ability of enzymes to be inhibited. So we start off with enzymes talking about what should happen in a normal reaction when they're proceeding, but what happens when you need to stop or slow a reaction and that enzyme is present? So maybe we have constitutive expression and that enzyme is always around, but we don't always want it to be active. So in the first, we have competitive inhibition. Competitive inhibition means that two things are directly competing with each other. So I have the substrate that wants to bind to the enzyme to undergo a reaction, and I have another that is competing with it. So this is here an example where they have the exact same shape that interacts with the active site. So they're able to both block that active site, and they can then stop or slow the reaction. Now, we have to remember that just because we're talking about inhibition, this is not necessarily a bad thing. There are times and places for reactions to occur, and there may be reasons in cells why you want that reaction's rate slow. The next is allosteric inhibition. So allosteric inhibition is happening when you have binding to a regulatory or allosteric site, and it blocks the active site in this indirect pattern. So here what we have is the substrate here is binding, looking for my picture. Oh, this got cut off on the side of my view here for a second. Sorry about that. So we have a molecule here shown this kind of blue Y-shaped structure that is binding to this regulatory site. And in doing so, the shape of the protein is changing. And so when it's changed, the substrate that wants to normally bind to the enzyme can no longer bind appropriately. Now, when that molecule, this Y-shaped structure, is removed, that is no longer a problem. It's able to go back to its normal set of conditions. So one of the things I've added here is often using a negative feedback system. So we can see here that when certain products are available, that will block that site. So if we have plenty here of these individual products, we don't need to keep breaking this substrate into the two halves. And so we have this negative feedback system. Non-competitive inhibition here is when our inhibitor molecule binds to the entire enzyme substrate complex and completely blocks it. So this one I'm thinking of kind of this wall, this complete block or barrier to this from proceeding versus these very systematic, these very specific bindings that occur with these other molecules. So we mentioned before that the goal of enzymes is to minimize how much energy has to be used. So we lower our activation energy, we lower the amount of energy that has to be used to conduct a particular reaction. And when we talk about energy, we can think about various forms of energy, thermal energy, radiant energy, electrical energy, mechanical, atomic, chemical. And in cells, we tend to think about this not in terms of where the source of that has come from, but in terms of the metabolic currency that we call ATP. And hopefully we have a little bit of a memory as to what ATP is. So in cells, ATP is this major currency. And again, if you need a quick refresher, ATP is named for adenine, the nucleotide base, and the triphosphate group that's attached. And then the central structure here that's holding that together is this ribose sugar. So an adenine nucleotide, a ribose sugar, and an adenine triphosphate. So we have these ATP molecules that are being generated in our cells by exergonic reactions, by those that produce or release energy. In contrast, there are many reactions in our cells that are not favorable, that require the use of energy or they consume that energy, and those are called endergonic reactions. Often in our cells, these are coupled together to make processes more energetically favorable and to allow us to drive these cellular processes of our microbial cells. In order to synthesize ATP, you probably remember learning about this a little bit. There are a couple of major mechanisms. We have substrate level phosphorylation, which just means you're adding a substrate, which is here a phosphate group, to this precursor molecule as ADP, using our enzyme here, our ATP synthase molecule, to make our ATP. So phosphate, ADP, so pretty much what you're saying here is 1 + 2 equals tri equals 3. Oxidative phosphorylation is when we use a series of redox reactions. So we couple reduction and oxidation reactions together. And we do this during the major respiratory pathways of our cells. And photophosphorylation is what we do in photosynthetic organisms. So we use the energy of the sunlight, we couple the energy of the sunlight to drive the formation of sugars. So we have 3 mechanisms for ATP. We have phosphorylation using ADP plus P equals ATP, oxidative phosphorylation, which uses redox reactions in respiration, and photophosphorylation, which allows us to use sunlight energy to undergo adding phosphates in our photosynthesis process. So overall, when we study these processes in cells, we call this idea bioenergetics, the idea that we have cellular energy that has to be added and released. So we have all these bulk processes, all these mechanisms that come together. The 3 major catabolic processes of our cells, those that take large molecules and break them down into smaller usable portions by our cells, are glycolysis, Krebs cycle, and the respiratory chain, or the electron transport chain. Now, this is the same as what we would talk about as the 3 core steps in a typical eukaryotic cell, even if it was a human or something higher up. So we want to not just talk about what happens, but try to highlight a couple of differences that are going to exist between microbes that are prokaryotic and eukaryotic compared to maybe some of the things we're used to. So the 3 core strategies, again, there are 3 core metabolic strategies that we're going to see in our microbes, which are aerobic respiration. So aerobic respiration couples together glycolysis, the Krebs cycle, and the respiratory chain, or the electron transport chain. Anaerobic respiration, which also uses glycolysis and the Krebs cycle, but instead during electron transport or the respiratory chain, it does not use molecular oxygen and uses a different final electron acceptor. And fermentation, which also uses glycolysis, but instead of going through a typical Krebs cycle and electron transport chain, we have a different series of electron acceptors that are used. And this is a much shorter process with a lot lower energy generation. So let's look a little bit at aerobic respiration. So we remember that glycolysis is always going to start with a glucose molecule. A glucose molecule is a 6-carbon molecule. The goal of glycolysis is to go through a series of oxidations with the end goal to be that we'll form 2 3-carbon pyruvate or pyruvic acid or pyruvate molecules. In this process, we're also going to generate small amounts of ATP and electron carriers called NADH. From that process, depending on what we're looking at, NADH can be used in electron transport chain, and it may also be coupled with other processes as well. The Krebs cycle, also called the TCA cycle or the citric acid cycle, is where we're going to then take those pyruvates or pyruvic acid, and our goal here is going to be to go through a series of steps to produce CO2, ATP, NADH and FADH2 as shown in the figure. Those are all, again, going to give us the ability to carry electrons that are going to move to the electron transport chain. And finally, here we have the electron transport chain that is going to accept those electrons being transmitted by NADH and FADH that we saw above, or FADH. And we're going to bring them through a series of electron carriers, through a series of sequential redox reactions, and going through that process that we defined before as oxidative phosphorylation, so that redox reactions chain, to be able to get to the point where we generate a hydrogen gradient and power ATP synthase to make ATP. Now, all of these processes are incredibly similar when you're doing a quick overview and comparing them between prokaryotes and eukaryotes, and between microbes and non-microscopic organisms. The couple of things that we want to keep in mind would be some core differences between prokaryotes and eukaryotes. So glycolysis is vastly going to be identical. Glycolysis is one of the unifying linking processes of nature. It is done by nearly every cell. The reason for that is that it doesn't require the presence of oxygen, which means it's an ancient process. It might be one of the oldest reactions that still occurs from early forms of life. So the only tiny difference with location for this is that the tail end of glycolysis, if we're going to be in a eukaryotic system, is going to end in the mitochondria. So we start in the cytoplasm, we go to the mitochondrion, versus if we're in a prokaryote, the entire process would be in the cytoplasm. There's no bringing the pyruvate to another source, another location, because there isn't another location to go to. Moving into the Krebs cycle. The Krebs cycle is vastly also going to be the same. The big thing we'd want to highlight here is that the Krebs cycle is going to occur in the cytoplasm entirely of a prokaryote, and the Krebs cycle of a so there, sorry for the break, pause there. So the Krebs cycle in prokaryotes is going to occur entirely within the cytoplasm, within the cytosol, whereas the Krebs cycle of eukaryote is going to occur within the mitochondrion. We want to remind ourselves, of course, that prokaryotes don't have mitochondria, so that's going to provide one of the really key differences between these organisms. The last difference that we want to point out with the electron transport chain shouldn't surprise us, because electron transport chain also occurs within the mitochondrium, and we don't have that structure within prokaryotes. So let's take an opportunity to do a comparison. I didn't think I had the time or the need to show this for each of those three steps, but I will show you the one here. So here is a zoom in of what process happens in the mitochondria. So here we are, this is the outer mitochondrial membrane, here's the inner membrane, so here's the intermembrane space. In a eukaryotic cell, we remember that electrons are traveling through these stages where they undergo those redox reactions. They're passed through, and as they're passed through this system, we have hydrogens that are being pumped into this intermembrane space, gathering this high concentration of hydrogens that will then flow through ATP synthase and power the ability here to convert to form ATP during something we call that chemiosmosis, or that idea that we're going to have hydrogens funneling down in to make ATP. So again, this is within the mitochondrial wall, within the inner intermembrane space. So what about prokaryotes? We don't have a mitochondria. What do we do? So notice here that where we actually are is not necessarily directly in the cytoplasm, but we are embedded here in the cell membranes. We kind of modified this procedure a little bit to give us a similar arrangement, but yet we're not anywhere near in the same spot. So notice here we've got the electrons that are being transmitted here from those carrier molecules. They're moving through the series of protein carriers. And then along the way, then we have hydrogens that are being pumped out of that cell to facilitate or balance out that difference in charge. And eventually the hydrogens are going to encounter ATP synthase. So we have the same name for this molecule, for this enzyme. And that is going to then be what powers us as hydrogens flow down through, causing ATP synthase to spin to phosphorylate ADP to ATP using here our inorganic phosphate. So the molecules, the proteins here are very similar. The nature of pumping out into a space is very similar, but here the periplasmic space compared to the intramembrane space, and the enzyme here is again the same. So when you're looking at these differences, when you're thinking about prokaryotes and eukaryotes and how we might see some differences because we focus more on prokaryotes and within bacteria in microbiology, this would be a really great thing to think back to as to why there are differences, because there are just huge structural differences between these cells. And then here's just one other view of that in case you wanted a little bit of a zoom in. So here is the cytoplasm, and here's outside here of the membrane. And you can see again here how the electrons are flowing through, just to give them in case you wanted to see a little bit more of the names and the structures. So in the electron transport chain, we remember here that this is going to be where we have our bulk generation of ATP. So this is a nice summary figure showing us those differences. In oxygenic or aerobic forms of this process, we would expect for the electrons to be obtained from splitting water, and then the electrons to be obtained here as we split through this molecule, and oxygen acting as the final electron acceptor. So at the end here of our electron transport chain, regardless of whether we are in a prokaryote or a eukaryote, we're going to generate around 34 ATPs of water and reform about 6 molecules of 34 ATPs and about 6 molecules of water. Along the way, if you remember, we also did have some generation of a couple of ADPs from the Krebs cycle and from glycolysis as well. And in addition, we generated those energy carriers, the NADH and the FADH, that were both used in the processes. So this is a nice summary equation for keeping track of what goes in and out. But for the micro standpoint, you want to know the bulk, you want to know the processes, you want to know in general what's made from them, where we obtain ATP from, what ATP is, and you want to understand how those structural differences between a prokaryote and a eukaryote will alter these processes slightly between the two systems. What part of the aerobic respiration releases CO2? Is it glycolysis, the Krebs cycle, the electron transport chain, or chemiosmosis? Take a second there and think about it. And hopefully you're saying, oh, I went back to my table, or I remembered from seeing it. And I remember that the Krebs cycle is a major place where CO2 is produced. Where does the electron transport chain occur in bacteria? In the cell wall, in the cell membrane, in the cell envelope, in the mitochondrial membrane, or 2 of the above are correct. Take a pause if you need to. You can physically pause the recording. So here's my cell membrane, which is where my process is occurring. And if we remember back, this entire structure, including the membrane, is called the cell envelope. So if the cell envelope includes the cell membrane, then 2 of the above are correct. Moving on to anaerobic respiration. I find that with anaerobic respiration and fermentation, sometimes these get a little less coverage in earlier biology classes. So if this isn't as much of a review for you, that's totally fine. With these processes here, we're really just focusing on what's going in, what's going out. If this was a cell bio class or a class that was really focused on the reactions, I would want to go through all the little working parts, but that's just really not the goal of this microbiology class. So if we're talking about anaerobic respiration, we remember that glycolysis is this unifying step. Very early prokaryotes on planet Earth underwent glycolysis. We expect for all of our microbes to do this. So in glycolysis, we're starting with glucose. And again, our goal is to make 2 pyruvic acids, 2 pyruvics, or oh my gosh, pyruvic acids or pyruvates, trying to combine the 2 words together. We then go into the Krebs cycle. Everything is the same here. We then go electron transport chain. Everything still seems to be about the same. The big difference that we get into is what's listed in red here as well, is that we have to have a different electron acceptor if there's no oxygen available. So we have SO42 minus, NO3 minus, CO3 minus. So it's our sulfates, our nitrates, our carbonates, these different options for possible electron acceptors. Most obligate anaerobes use that H plus that's generated and are able then to still have that reduction effect. And they are still able to synthesize large amounts of ATP, although aerobic respiration does tend to produce more ATPs than other forms. The form of bioenergetics and of metabolic strategy, I should say here, that was going to result in the least amount of energy produced is fermentation. So in fermentation, what are we still starting with? Glycolysis. Hopefully you immediately were like, I bet this has glycolysis in it too, because it does. And so after we have our pyruvic acid, here then we're going directly into fermentation. Now there are different types of fermentation. There are many different alcohols and acids and gases that can go into this. Two really common ones are lactic acid fermentation and ethanol fermentation, so alcohol fermentation. But really there's a ton of different products that can go into this. Now, fermentation, whether it's in a human model with lactic acid fermentation being a last ditch effort by your muscle cells to get a little bit of ATP, or as a major metabolic strategy, are just not going to produce a ton of ATP. Now, if a cell's needs are relatively small, or if it's a small simple cell, that might be okay. The larger organisms often cannot survive entirely on fermentation. We know that different organisms are really well adapted to particular forms of fermentation. And within microbiology, we encounter a lot more of these, probably because of the fact that we have microbes that have really diverse strategies for life. So let's look at some examples besides what's listed here that might be kind of interesting. So the first thing we've got here is a yeast system. So we can see that the yeast system has this idea again where you have the pyruvic acid, but we're able to embark on a process here using pyruvic acid to form acetaldehyde to make ethyl alcohol. So this is an alcohol fermentation step with yeast. Shouldn't be too surprising to you that yeast may produce ethyl alcohol as a byproduct of a reaction, because of course that's the system that beer and wine are set upon. Another really important one that I mentioned before is lactic acid fermentation. So we see this with homolactic bacteria, and also, as I mentioned, within human muscle cells as a effort when we run out of oxygen and we still need to be able to produce some ATP to keep going. So pyruvic acid here eventually being converted to form lactic acid in this anaerobic procedure. Now, there are so many different forms of fermentation that exist. Again, we mentioned yeast undergoing alcohol fermentation to make ethanol. And we mentioned here lactic acid fermentation by different forms like lactobacilli and streptococcus is another example. We have tons of different, we have formic acid, we have acetic acid, we have butanediol, propionic acid, butyric acid, all different forms. Now, what I hope you notice here is what types of organisms are the ones that are undergoing these in diverse forms of fermentation? Well, it's Enterobacter, Propionibacterium, Clostridium, Proteus, Streptococcus, Lactobacillus, Acetobacterium, yeast, Escherichia, Shigella. These are microbes. These are processes that have been that have evolved in microbes over millennia to serve and live in these diverse environments that they do. So this is more diverse in microbes compared to animals for many reasons. One, because bacteria live in more diverse environments, because bacteria have lower nutritional and energetic needs. Those are probably the two biggest ones I can think of, but definitely focus on, diversity here and what the actual needs of the organism are and why these strategies might be more common than in bacteria. So here is a comparison table. I love me a good comparison table, looking at aerobic respiration, anaerobic metabolism, including fermentation, and the different pathways, electron acceptor products, and strategies that are involved for a nice overview. Another connection I want to make here, going away from our processes that break molecules down into smaller ones, is processes that build things up. And so here what we're going to look at is photosynthesis. So photosynthesis is the source of chemical energy of life on Earth. We know we've got algae within our microbiology context. We're not necessarily focused on plants, but we're thinking about our unicellular and microscopic algae. We're thinking about our cyanobacteria, we're thinking about our green and purple bacteria. We're thinking about these organisms that are able to capture light energy and convert it into something that's usable by a large number of organisms on planet Earth. So you probably remember your equation, or remember seeing your equation for photosynthesis: 6 carbon dioxide plus 6 water, with the addition of light and the use of different photosynthetic pigments, allows us to produce glucose and oxygen. And oxygen production, of course, is critical because this built up over time on planet Earth, giving us the conditions that we have now. So as we've said many times, prokaryotes and archaea, microbes in general, are what gave rise to life on Earth, and not just because they were the first life forms, but because they transformed the Earth and made it into something usable for the organisms that are here now. We know that on land, plants are our primary photosynthetic organisms, but once we move into the water, once we move into the sea, that photosynthesis is occupied by algae, cyanobacteria, or green and purple sulfur bacteria, purple non-sulfur bacteria, these organisms, again, that are dominated by microscopic life and again transformed to seas and much of planet Earth. Most of the change, and we know too that of course all the first forms of life on Earth were aquatic, pretty much. So the transformation of the seas happened before the transformation of the land. So there are many different groups of phototrophic bacteria, those that use light, whether they're photosynthetic or a different form. I've mentioned a couple of these already. I tend to focus on cyanobacteria as a really classic example, but it's worth pointing out, as I've done a couple of times, that we also have the green sulfur bacteria. We have green non-sulfur bacteria, purple sulfur bacteria, and purple non-sulfur bacteria that all do possess photosynthetic pigments in some of their members. The most common forms of pigment are either going to be chlorophyll A, which is in cyanobacteria, which is part of the connection why we talk about cyanobacteria and modern plants, or that bacterial chlorophyll that I mentioned in the past lecture, which is structurally just modified from chlorophyll, but it has a different set of wavelengths that these organisms can absorb from. So they are different in that sense. Also, the cyanobacteria are the only oxygenic members. All the rest of the members are anoxygenic. And cyanobacteria are the only ones that split water. The others tend to use other forms of hydrogen sulfide, sulfur, hydrogen, or other forms of organic compounds. So looking at this, what you probably will realize that I've stated a little bit already is that cyanobacteria are closest to plants in every respect. So which chlorophyll is used, oxygen being used, water being the electron donor. And then these other forms, the green bacteria and the sulfur bacteria, tend to be, again, different form of chlorophyll, don't use oxygen. Sulfur forms of bacteria use sulfur as the electron donor predominantly. The non-sulfur bacteria use other organic compounds in place. So let's go ahead and just do a quick comparison of this, because again, there are some differences between the prokaryotic and eukaryotic system because of the structures that are present. So when you talk about photosynthesis, I think you probably remember to some extent that there are two major stages: the light-dependent reactions and the light-independent reactions. The goal of the light-dependent reactions is to take a water molecule and to split it. So you release oxygen when you split it, and you capture the electrons here that are present when you split them apart here from the hydrogen source. So in this process of light-dependent, your goal is going to be produce some ATP and some NADPH that can be used again as that carrier when we move into the light-independent. Those then are going to go through a series of steps to produce glucose, with the addition here of carbon dioxide, which of course is the carbon source. So we have the light dependent reactions where the predominant input is going to be water and sunlight. And we have the light independent reactions. The inputs are the materials from the light dependent and CO2. And the product that we're looking for is to produce glucose. So when we compare these in different types of organisms, here what we're going to do is look at a green algae, so a eukaryotic microbe, and a cyanobacteria, so a prokaryotic microbe that we talked a little bit already. Now remember, these are incredibly similar because they both use chlorophyll A, they both use oxygen, they both split water, so the processes are really similar. But the major difference here again is going to be where they occur. So if you're in a green alga, you are in a eukaryotic cell, which means that you are within the thylakoid, and the thylakoid is set here within. We zoom back out here. So if you go out, you'll see the thylakoid is stacked into a granum, and that granum stack is within this larger chloroplast, or specifically it's within the stroma of this chloroplast. So you can see the stroma is marked here in the figure. So we can see here in those light dependent, again, we need light, we need water, and those two components are coming in. They pass through a series of carriers, very similar to what we talked about with like the electron transport chain. They excite those electrons, they work through the system, hydrogen is being released. We go through that, hydrogens gather in the thylakoid lumen, they're captured and pushed out, ATP is produced as the ATP synthase spins, life is great. Now cyanobacteria, very, very similar, but this happens within the cytoplasm. So we have thylakoids, but they're contained within the cytoplasm, or often within these outer kind of folds here where the thylakoids are situated. And so within that thylakoid, we have our photosystem. So notice that we still have water coming in, oxygen coming out. We have the electron moving through this series of steps. We're accumulating hydrogen gradient on the inside of the lumen, and it's passing out through ATP synthase so we can generate more ATP. So is the process the same? Yes. Is the location the same? No. Again, because cyanobacteria are prokaryotic, they do not have organelles, they do not have, we go back here, they do not have chloroplasts. And so this is going to be not the exact same. So I think this is so interesting because you can have such diversity in terms of molecules, but somehow, even without having the same organelle, they've merged on the same process. It tells you how important this process is, how important these proteins are, because it's just so functional. The light independent reactions, we're going to encounter something very similar. So here we're in a cyanobacteria, and again, we're out in the cytoplasm here. The one thing that I want to highlight is that a lot of aquatic organisms, which a lot of these microbes that can undergo photosynthesis are aquatic, have to have a mechanism to, so what I want to say, to concentrate CO2. So we have this concentrating mechanism within here that we can build up our CO2 levels before we go into the Calvin cycle. So that's one major difference between a lot of these aquatic microbes and something like a regular land plant, is that we wouldn't expect for most land plants to have to accumulate CO2 because there's lots of CO2 surrounding them everywhere. So the rest of the cycle is the same. And again, I don't, for the sake of this class, need to refresh you on the major steps. But here we're looking at the major phases are carbon fixation, reduction and synthesis, and RUBP regeneration, which is our RUBP being the major, or Rubisco being the major driving force of this process. But again, our goal here is going to have to be to split these structures into different carbons and to then build them up into a series of 6 carbon molecules, every time building one additional 6 carbon molecule and continuing on with these intermediates to go through the rest. So the main job of chloroplast pigments during photosynthesis is to A, capture photons of light, B, absorb carbon dioxide, C, fix carbon dioxide into large organic molecules, or D, produce ATP. The main job here is going to be to capture the photons of light. Where do the light reactions occur in eukaryotic algae compared to prokaryotic cyanobacteria? A, both occur in chloroplasts. B, both occur in the thylakoid membrane. C, both occur in the cytoplasm. Or D, these cells are not photosynthetic. Take a second and think, or pause. And the answer here is both occur in thylakoid membranes. And I might want to caution against using the word membrane because it does kind of make it sound like I'm saying like an organelle membrane. inside the thylakoid, it would be a better way for you to say it. I shouldn't probably put the word membrane here in case it causes confusion. Okay, that's the end of the lecture topic. Again, from this, what I want is for us to have an understanding of what all of these bioenergetic processes are, what metabolism is in these cells, you know, and how the presence of organelles is going to alter these processes slightly, while still understanding that microbes still have to undergo basic life processes, even though they are smaller and less complex, and that these processes are vastly the same, which I think is kind of an amazing testament to how successful they really are. So we don't conserve something in biology because it's broken. We conserve it because it's full of great mechanisms. So for your check-in question, what I would like you to do is this a little bit of a elongated one. And I usually do 3 points for in class. I'm gonna put 5 points on this because it's longer and because you have to do it out of class. So this for me is a master study question, which is why I have it here. I've gotten a couple of questions like, how would you study, or how would you study for the upcoming exam? And this is the kind of stuff that I do. And as I got older, the things that I learned to do for myself, which was if I took an example, how could I apply the things that we talked about in class? Because it's pretty easy to memorize the definition or to memorize a factoid. But a lot of times class questions, and you will get some of these types of questions on exams, are application based. So what I want you to do is choose a microbe and list its levels of classification. I will post for you some options, but actually, you know what? I won't. If you want to go to a group of microbes, you can go back to the slide that we used previously in class that has that group of 24 different genre of microbes, if you want to find one from there. Otherwise, you're welcome to pick any microbe you want. I may add a link. We'll see if I decide that I can find a good source of a lot of random microbes. That'd be a really weird website. But it can be anything. It can be any type of microbe that you want. I would suggest, for the sake of filling out this sheet, that you choose a bacteria. I think that it'll fill in a lot easier, and you won't be stuck going, wait, does this eukaryotic cell have a particular shape or arrangement? But you have the option technically, but I would choose a bacteria. So what occupation or branch of microbiology may be interested in this microbe? So we'll say that you chose a bacteria and it's a prokaryote. We'll go to number 3. And you might say, well, is it health related? Okay, what branches of microbiology care about health? Is it agriculture related? Okay, which branches care about that? Does it spread in terms of like a communal infection? Okay, who would care about that? Is it responsible for making a food product? Like, is it related to one of those fermentation processes with dairy or with alcohol? Okay, what branch would care about that idea? Connected to one or more potentially of those ideas. You know, it may have some different components. 4, if it causes a disease, list it. What symptoms, treatments, vaccines, if any, exist? If you choose a non-pathogenic example, explain what impact it has in its environment instead. So if you choose something that's a mutualist with the human gut, well then, okay, what does it do for the human gut? Number 5, you believe your microbe of interest is present in mixed sample. How would you identify it? Trace through steps using any techniques you think would be helpful. So you may want to look up your microbe. Is there a particular way that people tend to identify it? So is there a particular type of media that's particularly useful to identify it? And of course, here I'm reflecting back on the 6 eyes, and that's what I'm expecting that you're going to trace through again, like we did in a past in-class activity. Number 6, describe the shape, arrangement, gram stain, and what the gram stain results mean about the morphology of the organism's cell wall. So again, I strongly suggest you pick a prokaryote because a eukaryote will not apply here. Add those components in. And again, let's say you choose a gram positive. What does that mean about the gram positive cell wall? If you choose gram negative, what does that mean about the gram negative cell wall? If you end up with a gram variable or a cell wall-less bacteria, you will have an additional comment there as well. How does the microbe obtain carbon and energy? Use the scientific terms. So here what I'm referring to is it heterotrophic? Is it autotrophic? Does it get its carbon from organic sources or inorganic sources? Add in those special words. So again, heterotroph, autotroph, chemotroph, phototroph, whatever you are using. Describe its niche using terms for temperature, oxygen, pH, and any other conditions that are appropriate for your example. So, if it doesn't live at a temperature that, let's say it's a human pathogen. Okay, well then it lives, in a mesophile condition. Let's say it's a human pathogen that requires oxygen. Okay, then it's an obligate arrow. Go through that list to explain its preferences. So I would expect there's no drawing or anything for this, so you should be able to type it all up and submit it. I will have a Dropbox on MO2 for you to do that. Sorry for dragging on here on this last slide. I'm just trying to anticipate what I think some of the questions might be, because you won't be able to ask them of me directly. I will, of course, be checking my email for this. And then I'll have this due on Sunday. But I would not advise you wait till Sunday. I would advise you do this right away on Friday, because you will have other studying that you want to do. And you don't want to feel like you have to complete assignments when you're trying to study. But I will give you the extra time since you were kind enough to give me a little bit of leeway here in presentation mode. So thank you again. I hope that you all have a great weekend and study hard. I'll post some additional announcements for upcoming materials. Take care, everybody.